Good morning, good afternoon, and good evening. Welcome back to Chapter 10. We're talking about viruses. We're going to move on, and we talked at the end of the last video, we were talking a little bit about mutation. Let's talk a little bit more about how mutations happen. And now this is not only just in viruses, this is mutations in general. A mutation is an alternation in DNA. The DNA has been changed. You can kind of think of it like sometimes you make a photocopy. Sometimes some of the letters get smudged and they're hard to read, and sometimes they're really hard to make out. That's sort of the same idea. When DNA gets copied, sometimes there are errors that happen, and when those errors get stuck in the copy, get left in the copy, now we have this thing called a mutation. There are a number of different kinds of mutations. One mutation would be a substitution. A substitution is when you put the wrong base in a particular spot. So if I'm making a copy of DNA, and this is an A, a T, C, G, T, T, right? If I make a copy of that side, I'm supposed to put a T here, and then an A, and then a G. Well, if there's an error, and this does sometimes happen, if I put a C there where there's supposed to be a G, I've now changed the code. This right here is a substitution. That's supposed to be a G, but I put a C there in the wrong spot. Sometimes you hit the wrong letter. That's a substitution, and that's a mutation. You can also have deletions and insertions. And a deletion and an insertion, I got a graphic coming in just a second. A deletion and insertion is where I just didn't include a particular letter, just missed it. Or an insertion is I put an extra letter in that wasn't supposed to be there. All of this means that the code is now written wrong. And if the code is written wrong, if I made a substitution, sometimes one bad amino acid will make a bad protein that still kind of works. If I have done a deletion or an insertion, it shifts what we call the reading frame, and I've now just made a protein that in no way can work. So here we see the code, AUG, right? Here's the code. This is all that matches. There's supposed to be a U here. Well, if I make an insertion where instead of putting a U there, oh, here it is. This is the insertion, an A. If I put the wrong letter in here, well, now I'm putting in the wrong amino acid. If I put one wrong amino acid in, sometimes that can lead to a protein that works poorly, but still works. If I delete a letter, deleting a letter means now everything downstream is read wrong. Because if it was supposed to be T, A, T, T, and then, all these different ones, by deleting one letter, T, A, G, G, C, G, C, A, T. I'm reading these, remember, 3 at a time. If I delete a letter, well, now I have to pull the next one in. It's not just that it's not there. It's that the whole thing just got pulled up one letter. And so now everything downstream from there, I am off on the stream. I am pulling one from the last one. So I didn't just make one wrong amino acid, everything downstream from there. And the same thing happens with an insertion. If I add an extra letter, that's not supposed to be there. I've now shifted the reading frame and everything downstream from there is wrong. This is what happens when you have a mutation. And most of the time, this is terrible. Most of the time, this just leads to death. But very, very rarely, it can lead to a benefit. If you think about it, if only 1 in a million mutations are beneficial, well, how many people are on the planet? Well, there's 7.5 billion people on the planet. If there's 1 in a million good mutations out there, that means there's 7,500 people out there that have a good mutation. Mutations, most of the time, are bad, but that's where the genetic change comes from. The source of genetic change comes from mutations. As I said, very few mutations are beneficial, but given enough time, enough mutations will lead to new species. Excuse me. Enough mutations will give it enough species. And given enough time, I'm not talking about 10 years, I'm not talking about 20 years, I'm not even talking about 100 years. I'm talking like 10,000 years. I'm talking like 100,000 years. Given enough time, enough of these random changes will lead to new species. This is how you get new species. Accumulation of mutations. We can also force mutations with something called genetic engineering. Genetic engineering is where we use science to change the instructions. We pull out the letters and we put in new letters. We change an A for a C. We change a U for a T. Genetic engineering, making new species. One of the ways in which we can do this is we take these things called restriction enzymes. Restriction enzymes are found in bacteria. That picture that we looked at quite a while ago that we showed how a virus infects a cell, that was specifically a virus infecting a bacterial cell. Bacteria have been infected by viruses for literally billions of years, and so to protect themselves, they make these things called restriction enzymes. And what they do is they look for certain sequences of DNA, and then they start to cut that DNA. So you find a particular gene that you want to use. You use a restriction enzyme to chop up the DNA, and then you hope that you can pull that gene that you want out and put it into another species, putting genes into other species. That's called genetic engineering. We did it with insulin. It was probably one of the first things we ever did it with. We pulled insulin genes out of a human, and we put it into a bacterium, and now the bacteria make insulin. Any diabetic, any insulin-dependent diabetic for the last really 30 or 40 years, the insulin that they have been using have been made by bacteria. Bacteria will easily pick up any free DNA. It's one of the cool things about bacteria, but also kind of one of the scary things about bacteria. And that is that any DNA that they find just floating in the environment, they'll pick it up. And the thing about bacteria is all the genes they have, they follow the recipe. They follow the instructions. They use all the genes they have. They don't just turn bits and pieces of them off like humans do. We use some cells will use some genes, other cells use other genes. Bacteria use all the ones they have. So if you give a bacterium a new gene, it just copies it, or rather it just uses it. It just makes the instructions, or excuse me, it just follows the instructions and makes the product. That's how you can make bacteria make insulin. If a bacterium is given a new little section of DNA, it's called a plasmid. Remember we showed before that the bacterial DNA is a big circle. It's a big circle, it's chromosome. A small little chromosome is called a plasmid. There are three ways in which bacteria can pick up new DNA. The first is called transformation, and that is they just find it floating around in the environment, and they do this all the time. Bacteria find DNA floating around in the environment, and they just suck it up, and now they're a new bacterial species because they picked up new genes and can make new products. This is how you get antibiotic-resistant bacteria. Rather, this is how you get multi-antibiotic-resistant bacteria. You got one person infected with two bacteria. You give them an antibiotic that kills off one of them that was resistant to a different antibiotic. And now the ones that survive that are resistant to the antibiotic you're giving them, they find that DNA floating around, they suck it up, and now that they've sucked up this DNA from this dead bacterium that was resistant to a different antibiotic, well, now this other species is resistant to both. Transformation. Next is transduction, and that is when a bacterium is given a piece of DNA from a virus. We do this quite often in genetic engineering, is we will package a particular gene that we want to give to a bacterium into a virus so that we know that the virus will give the DNA to the bacterium. Remember, the process of packaging viral DNA is really messy, and accidents happen all the time. So it's really easy to put a different kind of DNA in there. The last is called conjugation, and conjugation is called bacteria sex. Let's just kind of look at the different stages, and we'll talk about conjugation when we get there. Here's a little piece of DNA floating around in the environment. This bacterium finds it, and it just sucks it up. And this is called transformation. When a virus that holds a piece of DNA sits on a bacterium, it will inject that DNA into the bacterium. If that DNA isn't viral DNA, if it's from some other species, you just gave the bacteria a new set of instructions it never had before, and it'll just start following those directions. Again, kind of a cool thing about bacteria. They just follow the directions. Last is called conjugation. And conjugation is, there is this special little plasmid called the F plasmid. F stands for fertility. The F plasmid allows bacteria that contain the F plasmid to make these what we call sex pili, or called the F pilus. When one of these encounters another bacterium, it makes this mating bridge. And now that it's made this bridge, it will then start to send across a copy of its DNA. And the first thing that it sends across is the instructions on how to make these sex pili. It's kind of interesting that the ones that have the F pilus are referred to as male bacteria because they can make these sex pili. The ones that can't make them are called the female bacteria. What's the first thing that goes across when they have sex? The instructions on how to make the sex pili. And so the male and the female have sex, and what happens to the female? She becomes a male.